Good afternoon, one and all. I am Meenakshi Sundaram Palaniyappan. I am serving as the Project Director for the Center of Excellence in Innovations as well as the Head of the Department of Biotechnology, Agriculture College and Research Institute, Madurai. In today's topic, we are going to discuss about secondary metabolite production and in vitro germplasm conservation. So, to begin with, let us discuss about secondary metabolite production. Before we discuss how secondary metabolites are produced, we need to define the terms metabolite and secondary metabolites. As you all know, all plant based chemical compounds can be divided into two groups, namely the primary metabolite and the secondary metabolite. So, what is this primary metabolite? Please remember, the metabolites are those chemicals that are involved in both anabolic and catabolic pathways and that are needed for minute to minute and day to day essential activities undergoing inside the living cells. On the other hand, secondary metabolites are those substances that are not needed for the regular routine cellular activities. They are produced only under certain conditions. Primary metabolites include carbohydrates, amino acids, fatty acids and metabolic intermediates. On the other hand, secondary metabolites include alkaloids, terpenoids, phenols, essential oils and so on. So, we need to learn about these secondary metabolites in detail, but the detailed study on these metabolites are beyond the scope of this lecture, but still I will try to cover the basics of these secondary metabolites. To begin with, we will discuss about alkaloids. Alkaloids are those substances that contain nitrogen in their chemical structure. These nitrogen atoms, they are present in the aromatic rings like structures and they are normally derived from amino acids. So, alkaloids are derived from amino acids. As amino acids contain the amino group, nitrogen is passed on to these molecules. These nitrogen atoms, the amino groups, presence of these amino groups causes basic nature, provides basic nature to these secondary metabolites. So, especially these alkaloids. So, these are stored in the acidic vacuoles. So, during tissue culture, if our aim is to produce secondary metabolites, we should know where these metabolites are stored. So, alkaloids being basic in nature, they are stored in the acidic vacuoles. They are classified based on the heterocyclic rings and they are classified into many like piperidine, pyridine or pyrrolidine, tropane, phenolin, isoquinoline and purine alkaloids. So, an example for piperidine alkaloid is conine and an example for purine alkaloid is caffeine, the important constituent of both coffee and tea. These alkaloids are derived, as I told you earlier, they are derived from amino acids. For example, in this picture, different amino acids which act as precursors for these alkaloids are given here. To cite an example, lysine produces conine, tryptophan produces quinine. Quinine, as you all know, it is used for treating malaria. So, hope you understand what is the use of these secondary metabolites. Purine alkaloid is derived, especially caffeine is derived from aspartic acid, glycine as well as glutamine. So, the different amino acids that are present in a cell, they are acting as precursors for these molecules. These images are those plants from which these secondary alkaloids are extracted for commercial purposes. Now, we will move on to the next group, namely the terpenoids. They are also called isoprenoids because they form from a basic 5 carbon unit called the isoprene, a 5 carbon alkene called isoprene. This is the chemical structure of isoprene and these isoprene molecules produce an active molecule called isopentenyl pyrophosphate. So, this isopentenyl pyrophosphate acts as a precursor for the synthesis of different groups of isoprenoids. This isopentenyl pyrophosphate or the active isoprene molecule is synthesized in two different pathways, namely the acetate mevalonate pathway, which happens in the cytosol of a cell, as well as DOX-P pathway that happens in the plastids. So, acetate mevalonate pathway and DOX-P pathway are the sources for the precursor isopentenyl pyrophosphate. And this isopentenyl pyrophosphate, remember, it is a 5 carbon molecule. So, when condensation reaction happens between these isoprenes, new chemical compounds in multiples of 5 is what we are going to get. So, for example, in this pathway, isopentenyl pyrophosphate condenses with its isomer called dimethyl allyl pyrophosphate to give a molecule called geranyl pyrophosphate. This is the starting chemical reaction that is observed in a carotenoid biosynthetic pathway. Hope now you can understand that carotenoids are isoprenoids, but still these terpenoids can be grouped into different classes based on the number of carbon molecules present. Hemiterpenes, they are 5 carbon molecules. Monoterpenes, they are 10 carbon molecules. Sesquiterpenes, they are 15 carbon molecules. Diterpenes contain 20 carbons. Triterpene contains 30 carbons and tetraterpenes contain 40 carbons. Monoterpenes and sesquiterpenes, they make up most of the plant volatile compounds, whereas carotenoid pigments or the coloring pigments, they belong to the class tetraterpenes. So, this image, the image on the top gives you examples of the isoprenoids that are being synthesized, examples of menthol, geraniol and linalool. They are synthesized through the carotenoid biosynthetic pathway. And please remember, this carotenoid biosynthetic pathway gains significance in one of the biotechnological applications. It is the production of golden rice. So, you will be learning about how golden rice has been synthesized, for which a knowledge on the carotenoid biosynthetic pathway is an essential one. So, this table gives you the different groups of isoprenoids and their uses. This table contains 4 columns. The first column tells you about the precursor, the second column tells you about the class of the isoprenoid. An example of each class is given in the third column, and the fourth column tells you about the function. For example, the monoterpene, pinene and linalool, they act as defense substances, whereas tetraterpenes, they are example is carotenoids and they are involved in photosynthesis. Besides these, cholesterol, casein, gibberellin, a plant hormone, they also belong to terpenoids. Now, we move on to the third class of secondary metabolites, namely phenols. They include flavonoids, anthocyanins, flavones as well as flavonols. Please consult a good biochemistry textbook to have a enhanced knowledge on these molecules. These flavones, flavonols and all your phenols, they are produced through the shikimic acid pathway. So, definitely if we are working towards secondary metabolite production, a knowledge on the biochemical pathway is a must. So, some examples of phenyl propanoids or phenols include coumarins, lignans, lignin, suberin, cutin, stilbenes, flavonoids and tannins. Lignans are found in one of the important medicinal plants called Chila nelli. Hope you know what Chila nelli is. Does a bell ring? So, Chila nelli extracts are used for curing jaundice. Remember, secondary metabolites, they have multifaceted uses. So, that is the reason why we need to learn in detail about the biochemical pathways, so that we can exploit these secondary metabolites to the fullest. These secondary metabolites has several functions. They are involved in plant defense mechanisms. They also act as they are also produced through plant root exudates. They also act as natural natural attractants. They have some color. When we say plant defense mechanisms, we have to appreciate that plants are also living entities. They are also subject to the attack of microbes, that is pests and diseases. So, when a pest or a pathogen attacks, the plants produce compounds called phytoalexins. They are the defense compounds. They help the plant to overcome these pathogen or pest attack. Examples include isoprenoids, flavonoids and stilbenes. Some of the plants, they exude chemicals. They are acting as bacteriostatic compounds. Examples of bacteriostatic compounds include coumaric acid, 3-indole propionic acid and para-hydroxyl benzoate. As we all know, all flowers are colored, right? They are attractive in nature. These are all because of these carotenoid pigments, and they help us in the natural pollination events. Without pollination, no agricultural or horticultural produces are possible. So, these secondary metabolites are used in diverse industries. They are very good for as nutraceuticals. They are used in pharmaceutical industries. They are used as food colorants also. So, if we are able to extract a novel phytochemical with any of these properties, definitely we can mint money. Okay? They are also used in perfumery. For example, geraniol is it's a wonderful fragrance that we get it from geranium. Now, we are coming to the actual topic for today's discussion, that is production of secondary metabolites. Remember, tissue culture not only aims for mass multiplication or germplasm conservation, it is also used for secondary metabolite production. So, these secondary metabolites are those chemical compounds that are produced only under harsh conditions like pest attack or drought or something. So, under normal conditions, these plants do not produce. So, this technology of secondary metabolite production is more suitable for application using the medicinal plants. So, when we talk about production of these secondary metabolites through tissue culture, it can happen at different levels. At the laboratory scale, we make use of mini fermenters, whereas at the industrial scale, we make use of large scale fermenters. At the laboratory scale, callus and cell suspension cultures are used, whereas and also hairy root cultures. At the industrial level, we make use of equipments, engineered tools called bioreactors. So, to begin with the laboratory scale production, we need to understand callus culture. I hope callus culture has been discussed earlier. To briefly explain, we take a piece of a explant, a piece of a tissue that is called an explant. Here in this case, we take the carrot tissue, carrots, from which we try to harvest a vegetative tissue, and this tissue is placed on a nutrient media, and this nutrient media forces dedifferentiation so that callus is synthesized. And this callus, when it is subcultured to a embryogenetic medium, we get embryoids and we get embryo and a plantlet. So, this is the conventional callus induction procedure, the purpose of callus induction. When it comes to secondary metabolite production, we need to divert the callus to a different media, that is a liquid media. Whatever happened or was explained in the previous slide, it happens in a solid medium. So, the callus will now be transferred to a liquid medium. As I have already told you, callus can be classified into friable as well as hard callus based on the ability to grow into structures like embryos or some other structures. We call them as embryogenic callus or nodular callus. So, for secondary metabolite production, we need only friable callus, other calluses are not needed. So, the friable callus is taken and they are subjected to suspension culture, which has been already discussed. Suspension culture is nothing but you are trying to suspend the callus in a liquid medium, keep it under constant agitation so that cellular aggregation does not happen. The cells, they move away from one another and they start dividing. So, during suspension culture, cells or groups of cells are suspended in liquid medium, and this medium is supplemented with growth regulators like 2,4-D. 2,4-D is the auxin that promotes cell division. Culture is constantly agitated at 10 to 250 rpm, and the cells which are always in suspension, they grow faster than on solid medium. Just by putting them under suspension culture does not yield us secondary metabolites. We need to add some compounds called elicitors. These elicitors force these cells to produce secondary metabolites, and these secondary metabolites have to be analyzed whether our target molecule has been produced or not. So, whatever the growth parameters have been given to this culture has to be noted so that it is repeatable. So, noting down of all the growth parameters like temperature, agitation, what is the chemical that has been used, what is the hormonal concentration, etc., has to be thoroughly maintained, thoroughly recorded. It also has to be thoroughly investigated so that we achieve maximum production of our target metabolite. So, to optimize this secondary metabolite production in vitro, we have to establish suspension cultures that are going to produce a content of our required phytochemicals. So, once we are able to identify, we have to select the highly productive clones among the cultures available. You have to optimize the culture conditions, and this table tells us about the different types of secondary metabolites that have been produced either through callus culture or suspension culture. This table has 4 columns: compound in the first column, plant species in the second column, and the third column tells you about the yields and the different types of culture and the plants, and the last column tells you about the culture types. For example, ginsenoside has been synthesized from Panax ginseng. So, when you culture using callus, we get 27% dry weight of ginsenoside. On the other hand, harvesting ginsenoside from the plants themselves gives you only 4% of yield. So, this table tells you the potential of callus culture as well as cell suspension culture in secondary metabolite production. Not only callus culture, hairy root cultures are also very good for secondary metabolic production. So, first we have to understand what is meant by hairy roots. So, for secondary metabolite, we use a leaf or a stem bit or some other vegetative tissue as a explant. Even reproductive tissues also can be tried. So, when these explants are infected by a bacterium called Agrobacterium rhizogenes, the RI plasmid present in this bacteria induces the production of hairy roots. So, the plants, instead of producing adventitious shoots or roots, they are forced to produce hairy root like structures. This hairy root like structures, they are longer, they have larger surface area that promotes these secondary metabolites. And examples of secondary metabolites produced using this hairy root culture technique include Taxol, vinblastine and vincristin. I hope everybody remembers the usage of these molecules. They are all anti-cancer compounds. Taxol is obtained from Taxus brevifolia, and vinblastine and vincristin, they are obtained from Catharanthus roseus. So, this image tells you how we can induce hairy root cultures. So, on the top left you have a leaf that has been subjected to infection by Agrobacterium tumefaciens. This leaf has been derived from in vitro cultured tissue, in vitro cultured plant, that is given at the left hand side bottom. On the right hand side top, we see a close up view of the hairy root cultures, and on the bottom right hand side we see large scale cultivation of these hairy root culture with the objective of producing secondary metabolites. As I said earlier, if we want to produce large amounts of these secondary metabolites at the industrial level, we need an engineered equipment called bioreactor. So, this bioreactor is defined as a device or an apparatus in which living organisms, especially bacteria, synthesize useful substances or break down harmful ones. So, whether it is anabolism or catabolism, bioreactors are helpful. They produce something useful. So, these bioreactors can be divided into two groups, namely submerged culture bioreactors and solid state bioreactors. Submerged culture is identical to that of your suspension cultures. So, these submerged culture bioreactors, they are used for producing secondary metabolites, anabolic pathway is there, whereas the solid state bioreactors, they are used for degradation of harmful catabolites. So, it is a catabolic pathway. Examples of submerged culture bioreactors include stirred tank bioreactors, airlift bioreactors and bubble column bioreactor. Examples of solid state bioreactor include tray bioreactors, packed bed bioreactor, rotating drum bioreactor, agitated and forcibly aerated bioreactors. Explaining the functions, the mechanism by which they are constructed, they are operating is again beyond the scope of this lecture, but still I will try to show you some images of these submerged culture bioreactors. So, the first one, before I show you the images, we need to understand what are the factors that have to be manipulated so, so that we are able or to produce efficiently the target phytochemicals. The factors include the gaseous atmosphere in which these cells are present, the level of oxygen, the level of carbon dioxide, the pH of the media, the minerals that are present in the medium, sugars, hormones and plant growth regulators, liquid medium rheology as well as the optimum cell density. These parameters have to be standardized at the laboratory level, but the same thing will not work at the industrial level. So, definitely a scientist has to join hands with the biomedical engineer, so that these factors are exactly replicated on a large scale. So, these bioreactors, they are operated in different modes. Some of them they operate in batch mode, some of them they operate in fed batch and some of them are continuously fed. So, the mode of operation, how the phytochemicals are harvested, again is beyond the scope of this lecture. So, this diagram tells you how the medium is kept under constant agitation, so that there is the cells do not aggregate, the pH is uniformly maintained, and how the gases are circulated to promote the production of secondary metabolites. The construction of the different types of bioreactors, it dictates how the medium is mixed. So, you have on the top left you have stirred tank reactor, so where you have impellers to mix. In a bubble column reactor you have sparges for forcing air through the bioreactor. In an airlift reactor we make use of draught tubes, and in airlift reactors we also use outer loop mechanism. The images that are present at the bottom, rotating drum reactor and membrane reactor, they are for degradation. They are used for degrading contaminating waste substances. So, this is a diagram of a stirred tank bioreactor. So, you have different parts, mechanical parts that are going to be operated. This can be operated in a batch mode, fed batch or continuous mode. To understand the magnitude of industrial level production using a stirred tank bioreactor, this image is given. I hope you understand what the size of the reactor is. The stirred tank bioreactors become operational only when a protocol is standardized at the laboratory level. So, at the laboratory level, we begin with suspension cultures in a small scale, which is converted to, or it is extrapolated to, a benchtop bioreactor. Then we have a pilot scale bioreactor, then you have a industrial scale operation. So, the image on the top right hand side, you have a 15-liter bioreactor which is at the laboratory level. At the bottom you have bioreactors of capacities 150 liters as well as 1500 liters. See the complexity. So, we should not work as individuals, we have to work as a team, so that we are successful. So, this image tells you about airlift bioreactors. I am not going to explain much on them. This is the actual airlift bioreactor. This is an image of a bubble column bioreactor that has a sparger, and this table tells you about the different bioreactors that have been used for producing these molecules, secondary metabolites at the industrial level. So, this table has 4 columns. The first column tells you the name of the plants. The second column tells you about the products of the name of the secondary metabolites. The third column tells you about the bioreactor type, capacity and mode of cultivation, and the last one tells you about the product quantity. For example, in Ancusa officinalis, rosmarinic acid has been synthesized using 2.5 liters batch type stirred tank bioreactors, and they were able to produce 3.5 grams per liter. This table follows. So, you have multiple examples of secondary metabolites. Now, we are moving on to a novel type of bioreactor called a photobioreactor, which makes use of sunlight as the energy driving force. So, we try to grow moss or cyanobacteria. It is used for producing terpenoids. So, the image tells you how sunlight is used. So, the moss or your cyanobacteria, they are grown in polybags and they are maintained like this. The next one is algal bioreactor, which is used for producing nutraceutical grade algae. These algae are used in health, food as well as biofuel market. So, to sum up, the secondary metabolites are important molecules of commercial importance. They help us cure diseases, they act as pheromones and what not. So, you have plenty of opportunities because this area is relatively unexplored. If we are able to identify a couple of molecules, it is good, you can invest money so that you can establish a good biotechnology company. Now, we move on to the next topic, that is in vitro germplasm. So, this in vitro germplasm conservation is what we need to understand. But before I explain, we have to understand what is the meaning of germplasm. So, the term germplasm refers to the sum total of genes that are present in a particular species. Remember, for example, India is known for mangoes. You have thousands of varieties of mango, not only in India, across the globe. In the south, you have varieties like Alfonsa, you have varieties like Rajapalayam Sappatai, you have varieties like Kilimukku and so on. They are all mangoes, but they have their own distinct flavor, aroma and so on. So, what produces these specific attributes, it is all the genes. So, if we are able to contain or collect all the varieties with varied characteristics, and if we list out the genes, you call it as the germplasm of genes. Germplasm actually refers to growing diverse varieties of a single species in a single area, with the purpose of conserving the gene pool. So, germplasm conservation can be done either in situ or under ex situ conditions. So, in vitro germplasm is something that happens ex situ. In situ means as is where is conditions, and ex situ means from you are moving the plants to a different locality with the objective of conservation. So, ex situ conservation involves the use of seeds. Okay. You can conserve seeds, or you can take up in vitro conservation using meristems, embryos as well as shoots. So, if we learn meristem culture, shoot tip culture as well as embryo culture, we can take up in vitro germplasm conservation. The advantages of in vitro conservation include, you need a small space, say a 10 by 10 culture room is good for holding more than 1000 cultures. On the other hand, growing 1000 different plants, it is not possible in a 10 feet by 10 feet room. So, ex situ germplasm, in situ germplasms are used for conserving the real plants. In vitro conservation, we try to collect the explants and culture them. They will be safe because they are in a protected environment. Some cases embryo culture in mango as well as coconut enables you to conserve germplasm. It is also suitable for international exchange because the thumb rule of tissue culture is sterility. When it comes to international exchange, quarantine is a major issue which takes care of limiting the import of unwanted pests and diseases into a country. Since all these explants are grown under sterile conditions, quarantine issues will not matter. They can be stored for varied time periods from minimum to long duration. They can be stored for a short term, maybe for 4 to 8 weeks, medium term in months or years, and for long term unlimited period. I would like to highlight Svalbard facility here. Svalbard is a facility available in Norway, where all the germplasms of all the agricultural and horticultural crops are stored for ensuring self-sustenance to our future generations. So, the approaches of in vitro conservation include short term approach, medium term approach as well as long term approach. When we talk about short term approach, you can use any of the tissue culture techniques like embryo culture, shoot tip culture or meristem culture. You can grow the plants in the field, you can grow the plants under sterile conditions in the laboratory by repeated subculturing. With regards to medium term, we should make use of the facilities like cold storage, wherein culture conditions play a major role. So, the cultures have to be stored at non-freezing temperatures of 4 to 6 degrees centigrade, and the supply of oxygen matters. So, we have to grow them in a low oxygen environment so that we curtail the metabolism, so that the longevity is enhanced. Also, the medium that is used for culturing as well as the degree of desiccation matters when it comes to medium term storage. On the other hand, long term storage involves cryopreservation. As I mentioned in one of the lectures earlier, cryopreservation uses liquid nitrogen. So, the samples have to be pre-treated, the samples have to be pre-treated. The steps involved begins with the choice of the material. The samples have to be pre-treated so that they avoid or they overcome the freezing injury. Then you subject it to short term freezing and then you keep it for storage in a cryopreservation facility. Once storage is enough or once you want to recover the plants, thawing has to be taken up. So, before we attempt this cryopreservation technique for germplasm conservation, we should have standardized the in vitro culture protocols. Without a standard protocol for the target species, attempting cryopreservation is going to be a waste, because after thawing you have to successfully revive your plants. If you fail, you are going to lose your germplasm. So, it involves post treatment also. So, the cryopreservation begins with the choice of the material. The rule of the thumb is the material should be as young and as meristematic as possible, because meristematic cells, they have larger nucleus, they have a dense cytoplasm, they have small vacuole, they have very good capacity to withstand freezing. They can be sampled from in vivo or in vitro grown plants, and meristems can be used, shoots can be used, as well as somatic embryos can be used for preservation. As I told you, the tissues, they have to be pre-treated so that they can overcome the freezing injury. So, they are treated with chemical substances called cryoprotectants, for example, sucrose, mannitol, sorbitol, DMSO, polyethylene glycol are used as cryoprotectants. How these molecules confer freezing tolerance is not known. The mode of action is not clear. It is postulated that these molecules, they dehydrate the cells and they protect the membranes from freezing injury, but the actual mechanism is yet to be resolved. Freezing can happen at different speeds. Slow freezing can happen, rapid freezing can happen, as well as ultra rapid freezing can happen, which is also called the vitrification method. So, the methods vary, so it has to be standardized. So, this picture tells you how slow freezing happens. For example, a tissue is taken and it is treated with 0.5 molar DMSO, 1 molar sucrose and 0.5 molar glycerol on ice. They are treated for 1 hour, after which they are transferred to cryo vials, and they are frozen from 0 to minus 35 degrees centigrade at a rate of 1 degree Celsius reduction per minute. So, achieving or moving from 0 to 35 degrees takes a maximum of 3 hours, after which the samples are transferred to liquid nitrogen. So, from minus 35, the tissues are thrown into a temperature of minus 196 degrees centigrade, where they are held for 30 minutes and they are taken for storage for a longer time. Whenever the samples have to be revived, they are removed, they are plunged into a water bath at 40 degrees centigrade for rapid thawing, and they are recultured on a fresh medium. So, this flow chart tells you how the samples are prepared, stored, and how they are revived. Similar is the case in ultra rapid or vitrification method, so which I am not going to explain, it is self explanatory. So, these samples have to be stored in liquid nitrogen, hence called cryopreservation. So, we have to establish a facility with good supply of liquid nitrogen and good supply of liquid nitrogen canisters for storing these samples, else cryopreservation cannot be carried on. So, thawing has to be taken up once you are going to revive these plants from their culture. It could be slow or rapid. It has to be immersed in water, as I said earlier, at 40 degrees centigrade, and then it has to be transferred to a culture medium where the protocol has been already established. So, during post treatment what we have to do is we have to remove the cryoprotectants, otherwise they will interfere. So, before the after thawing and before it is transferred to the culture medium, the cryoprotectants have to be removed. So, we can change the medium because liquid nitrogen, it is liquid, when we transfer it to a culture medium it is going to be solid. So, we are altering the medium there. Once you transfer it, you need to ensure whether the cells are alive or not, for which we have to perform a viability assessment test. We use chemicals like fluorescein diacetate as well as triphenyl tetrazolium chloride. These are some of the chemicals that are capable of entering into live cells and they are getting catabolized. The color change that happens because of living cells tells us that the tissues are alive, and then you can take up culturing experiment, reviving of the cultures. Thank you.